Hello, everyone. I am so delighted to be here to share with you my ideas around deuterium and hydrogen-containing gases. I hope that some of you will be sufficiently intrigued by what I have to say to be willing to consider, design, and implement experiments that might either confirm or disprove my ideas. So my talk today is, are small hydrogen-containing gases molecules essential for maintaining low deuterium in mitochondrial water? I want to start with a quote, which I love. Hydrogen is a light, odorless gas, which, given enough time, turns into people. And this is Carl Sagan's version of a quote, which was attributed to the cosmologist Edward R. Harris. So this is an outline of my talk. You can see it's all about gases. Introduction, molecular hydrogen and methane, and then hydrogen peroxide, and then ammonia, and then hydrogen sulfide and dimethyl sulfide, and finally a conclusion. So the introduction, the big picture is that hydrogen gas, which is produced by gut microbes, probably has very little deuterium. And then the question is whether this is also true for other hydrogen-containing gases, which are very common in metabolism and have important roles to play. For example, ammonia, methane, hydrogen peroxide, hydrogen sulfide, and then dimethyl sulfide, a real sleeper there. Methane is converted to small organic molecules by gut microbes, and those molecules are likely also to be severely depleted in deuterium. Hydrogen peroxide, of course, is produced many parts of the cell, and in fact, outside the mitochondria, and then it can be imported into the mitochondria to bring in low deuterium protons. And which then it can be converted to water by glutathione peroxidase, which is highly expressed in the mitochondria. Ammonia handling in the body appears to focus on deuterium. This took me a long time to figure out because ammonia is complicated. Hopefully we'll have time to expose that. Hydrogen sulfide is metabolized in the mitochondria, and that releases protons that are likely not to be deuterons. Dimethyl sulfide, produced by microbes at river mouths, seeds clouds. So you guys are all familiar with this, mitochondrial ATP synthase. The protons are pumped into the intermembrane space, and then they crowd over here and they want to get back out because of the low amount of protons in the matrix. And they come through these ATPase pumps. And then if a deuteron happens to be in here, it will harm the pump and cause it to be less efficient at making ATP and also reduce producing reactive oxygen species. So this was a fascinating paper from 1961. I have not found anything later that also shows this, which is frustrating. But this gas was produced by a marine microbe using a hydrogenase enzyme that bore a great deal of resemblance to many hydrogenase enzymes that are expressed by the gut microbes. And this, quoting from this paper, mass spectrometric analyses of the hydrogen produced by growing cells showed a deuterium content of about 30 part per million. That's way down from 155, which is what is typical in seawater. And the same was true where the glucose or formate was a substrate. It can use metabolize glucose and formate into carbon dioxide and hydrogen gas. The intracellular water and cellular hydrogen were not significantly depleted, meaning that it was strictly the gas that had this effect. Now, it's interesting that microbial nickel iron hydrogenase has a very high kinetic isotope effect for deuterium extraction, as high as 43 in an acidic environment. So that is really significant. So this also makes a case for hydrogenase enzymes in the microbes being very important for producing hydrogen gas that's severely depleted in deuterium. I also found this very interesting article about methane, and this is in volcanic discharge. Extremely deuterium depleted methane revealed in high temperature volcanic gases. And it was naturally low in deuterium. And is it the fact that it's a gas that's causing it to have low deuterium is the question I ask. So these are some other molecules that come to mind when you think about this idea. You've got two hydrogens here, hydrogen peroxide, you've got two there. Methane has got 4 protons. Ammonia's got 3. Molecular hydrogen, of course, is nothing but hydrogen. And then you have dimethyl sulfide with 2 methyl groups that are possibly depleted in deuterium. And I think methylation pathways are also a quite interesting point. So might it be that deuterium tends to stay behind, dissolved in the aqueous medium for all these gases, primarily because it's twice as heavy? That's my question. So now first considering molecular hydrogen and methane, which are closely tied with the gut microbes. So the gut microbes produce abundant molecular hydrogen gas, and they use it to reduce carbon dioxide to small organic molecules, and particularly acetate, which then becomes butyrate, and propionate. All of these short chain fatty acids all come from this hydrogen gas and therefore are likely to be depleted in deuterium. And of course, butter is a low deuterium nutrient and it has a lot of butyrate. So that's just a hint that that might be the case. Methyl groups can be traced back to methane via formaldehyde. So formaldehyde is an intermediary between methane and methyl groups, which means that, and that's got two protons that are likely to be depleted in deuterium. So are the methylation pathways important? Specifically because they supply deuterium depleted nutrients to mitochondria systemically. That's my case I'm making. So a microbial derived metabolite of methyl groups, trimethylamine oxide, TMAO, may be a marker for deuterium overload in mitochondria. And I have a paper together with Laszlo Boros under review right now about this story, this potential idea that TMAO which is a risk factor for many diseases, and in fact, a causal factor for many diseases, could be a marker for microbial disruption in the gut associated with severe deuterium toxicity in the mitochondria. So what happens to the hydrogen gas? Here you have the hydrogen producing bacteria. They make hydrogen. Hydrogen is used as a reducing agent by these sulfate reducing bacteria, methanogens, and acetogenic bacteria, all of which produce fantastic products that are extremely useful to the host. So you have the methionine comes out of the sulfur pathway, universal methyl donor. This one, the methane it provides the important sources. Methane is an important source of NADH and H2O water with deuterium depletion, and also formaldehyde. And citric acid cycle here, acetate is acetyl coenzyme A, which feeds into the citric acid cycle. So all of these are really important in the metabolism. And then this just gives you some of the, bacterial products. Hydrogen gas, sulfate, carbon dioxide, and carbon dioxide, both here, methanogens and acetogens. These guys produce the butyrate and the acetate. Butyrate is fuel from the gods for the colonocytes. They love butyrate. 80% of their diet normally would be butyrate, and it's often deficient in association with diseases of the gut. And then methane becomes formaldehyde, which becomes methyl groups. And then this is the whole methylation pathway sequence, which becomes important, I suspect, because those methyl groups are low in deuterium. And then you have the sulfate producing hydrogen sulfide, which actually gets taken up by the mitochondria very aggressively. And the hydrogen is released, the protons are released into the medium. And so here we have methyl groups, which then become products through other pathways, methionine, choline, and L-carnitine. These are all very important nutrients that keep us healthy. And these two are found mostly in animal-based foods. So hydrogen gas is central to metabolism. I built this complicated picture here of all the different pathways. Choline and betaine and L-carnitine come through to produce the TMA, which then becomes TMAO through oxidation by the liver if it doesn't go through these pathways, which is what it needs to do if these guys are healthy. So the methylotropes, if those are not adequate in the gut, this becomes TMAO and causes trouble. But this comes through this pathway here and produces really good things like the methylation pathways here. And the methanogens can also produce methane, which can go through this pathway. So and then you have over here A. mucinophila, which is a very important beneficial microbe in the gut that actually eats the sulfomucins that are produced by the colonocytes. And so these small organic, and also dietary fiber comes in from bifidobacteria. These molecules are what become hydrogen gas by hydrogenotrophic bacteria. So you have all these different bacteria playing a role. So my hypothesis is that gut microbes recycle molecular hydrogen and then deplete deuterium through that. Colonocytes thrive on low deuterium butyrate. These goblet cells are the ones that produce the sulfomucins, which A. mucinophila consume and convert back into hydrogen gas. These guys release P9, which actually encourages the human host cells to release GLP-1. GLP-1 it curbs appetite. So we have an obesity problem epidemic in America. This hormone curbs appetite and promotes insulin release from the pancreatic beta cells. And GLP-1 is, of course, they have all these GLP-1 analogs that are very popular as drugs these days to help people lose weight. So TMAO is oxidized to TMAO. TMA is oxidized to TMAO in the liver whenever these anaerobic methylotropes are scarce, which can happen due to gut dysbiosis. Methyl groups support mitochondrial health. So molecular hydrogen miracle worker, this paper here really sings the praises of hydrogen gas and showing all these different beneficial effects that it has on the body. And some significant quotes from that paper: hydrogen may prevent inflammation and regulate the nutrient sensing mTOR system, autophagy, apoptosis, and mitochondria, which are all factors related to aging. Hydrogen can also be used for prevention and treatment of various aging-related diseases, such as neurodegenerative disorders, cardiovascular disease, pulmonary disease, diabetes, and cancer. So does TMAO signal deuterium overload? It's a risk factor for many chronic diseases. Interestingly enough, they've done experiments with deuterated choline and deuterated L-carnitine on mice and shown that they cause deuterated TMAO to show up in the blood. And the enzymes that metabolize TMAO in the gut, TMA in the gut, which will produce hydrogen gas, have a very high deuterium kinetic isotope effect. And so they recycle choline and L-carnitine by this anaerobic oxidation. And that's going to be a process also to strip out deuterium. Deuterated TMA cannot be metabolized by the gut microbes, so it ends up as TMAO. So when you have high TMAO, it could be a marker for a systemic deuterium overload. It's certainly a marker for gut dysbiosis, because if there are no gut microbes, you don't get any TMAO. It's definitely a product of gut microbial activity. So now my next topic is hydrogen peroxide. The big picture: hydrogen peroxide is produced in many parts of the cell, including and importantly in the external milieu. So a common metabolic pathway is from oxygen to superoxide to hydrogen peroxide to water. Hydrogen peroxide is water soluble, and it can cross lipid membranes without even any transporters, traveling from outside the cell or other parts of the cell to the mitochondria, where glutathione peroxidase is ready to convert it into 2 molecules of water. And that water is likely to be deuterium depleted. It may be an important aspect of hydrogen peroxide's role in the organism to supply mitochondria with deuterium depleted water. And so inflammation is a problem with many chronic diseases, which produces hydrogen peroxide. And that may be a process by which the body is trying to deliver low deuterium protons to the mitochondria. So this is just a figure that I found in this paper here, showing the importance of hydrogen peroxide in metabolism. And you can see you have a bunch of things outside the cell that produce hydrogen peroxide. These are the immune cells, the phagocytes. And there's a receptor ligand interaction that produces hydrogen peroxide. You also have NADPH oxidase, which is also called NOx, a very important enzyme, which actually produces superoxide outside the cell. It's inside the cell, but it sends out electrons to convert oxygen into superoxide. And I can show you here with some steps that I'm adding to this figure, which was not included in the original figure, but superoxide, NADPH oxidase produce superoxide, which goes to hydrogen peroxide through SOD3, superoxide dismutase, which is out there in the outside the cell. And then that gets brought into the cytoplasm. And that's a way to, these H2s are going to be low in deuterium, if I'm right, which means that we're bringing in low deuterium protons through this process. So these antioxidant enzymes play multiple key roles as sensors and regulators of signal transduction. There's a gazillion papers out there on antioxidant enzymes and hydrogen peroxide that induces diverse biological responses as a signaling molecule. The cysteine residues pick up on and act as redox switches, and then we have this SOD3 converting O2 minus H2O2 in the extracellular space, which then is allowing you to enrich the deuterium outside the cell because you're bringing in protons through the hydrogen peroxide process. You're bringing in protons that are not likely to be deuterons. The peroxisome is a source and sink of hydrogen peroxide. This was a lovely illustration of this, where you have the different color codes to show. On the left-hand side, you've got the hydrogen peroxide showing up inside the peroxisome and then going out into the cytoplasm. Or if you have too much on the outside, then it comes in. So because there's the membrane, it freely transports it, and even has transport proteins to encourage moving the hydrogen peroxide across the membrane. And so they're increasingly recognize these peroxisomes as important hubs in redox signaling. They facilitate the transport across membranes. Catalase is primarily localized in the peroxisome, and it converts hydrogen peroxide into water and oxygen. And then antioxidant enzymes act as peroxide sensors. So when the cytoplasm has excess hydrogen peroxide, the peroxisome imports it, which reduces its internal deuterium load, enriching the deuterium in the cytoplasm. When the peroxisome has excess hydrogen peroxide, it releases it into cytoplasm, and then the mitochondria take it up. So that's supplying from the peroxisome to the mitochondria, it's supplying deuterium-depleted protons. So aquaporin channels import. These are the channels that actively import hydrogen peroxide, and you can see there's several of them indicated here. These are all aquaporin channels. Here's your hydrogen peroxide outside the cell. There's your NOx, which is causing this thing that I showed you earlier with the SOD3, producing O2 minus, which becomes hydrogen peroxide, which gets inside the cell. And then once it's in the cytoplasm, it can move into these organelles and providing them with low deuterium protons. And of course, the mitochondria here and the mitochondria with can convert superoxide to hydrogen peroxide. And of course, they can also produce water from the hydrogen peroxide, which is not shown here. So the cell reduces its internal deuterium load by importing hydrogen peroxide. The organelles reduce their deuterium load by importing hydrogen peroxide, and then the aquaporins transport it across the membranes. The ER mitochondrial peroxisome triangle, redox triangle here, showing these three organelles, the mitochondria, the peroxisome, and the endoplasmic reticulum. This one also plays a role in providing the hydrogen peroxide that I think is then going to eventually make its way to the mitochondria. So they have this kind of triangle here where this is all coming in and coming into the mitochondria, which can then produce the water. So here's all the hydrogen peroxides, and then you've got these two waters over here in the ER and in the mitochondria. So again, the hydrogen peroxide is produced in the ER, the peroxisome, and the mitochondria, and glutathione peroxidase and peroxide redoxin can reduce it to water. So this whole process is going on all the time in the cell under conditions of stress of any sort. This whole pathway is upregulated, so the cell is trying to further reduce the deuterium load in its mitochondria. Lactic acid bacteria also produce abundant hydrogen peroxide, and here's a figure from this paper here. And you can see coming from glucose, you can get to acetate, and you can also get to lactate this way. Pyruvate to lactate oxidizes NADH to NAD+. When lactate is converted back to pyruvate, hydrogen peroxide is produced from oxygen. That's probably going to be low in deuterium. In an alternative pathway, pyruvate is converted to acetate, while both ATP and hydrogen peroxide are produced. So this is a useful pathway here for producing these metabolites that are going to be very useful in controlling hydrogen peroxide levels. My next topic is ammonia. The big picture, ammonia is a gas, but ammonium, NH4+, is an ion. It's dissolved in water. Only NH3 can get across the membranes naturally without any help. NH4+ needs to have special transporters to get it across membranes. The gut microbes metabolize urea, which is produced by the liver, and that releases ammonia in the gut, which is then transported back to the liver by these epithelial cells that line the gut. And they take up ammonia, but not NH4+. So you can notice when things are taking up ammonia, I suspected that those protons are likely not to be deuterons. If they take up NH4+, all bets are off because this is dissolved in the water. So the red blood cells also take up, very interestingly, I thought, ammonia, but not NH4+. And that ammonia acts as a storage depot for ammonia in the blood. It sort of keeps the ammonia levels in the blood manageable because ammonia will be toxic if it's too high. And then the red blood cells deliver that ammonia to the liver, which then converts it to the urea, which also goes back both to the general circulation and also back to the gut. So in the kidneys, complicated things happen with ammonia. We'll try to get into that a little bit more detail. They synthesize ammonia from glutamine, and they pass it through a complex process that appears to recycle ammonia as NH3 into the circulation and then export ammonium through the urine. So there's a whole process of distillation there where the ammonia gets captured by the red blood cells and the ammonium gets released in the urine. So this is just showing you that ammonia readily passes through the plasma membrane. In the extracellular environment, if you have a lot of ammonia, it will just seep through across the membrane and eventually accumulate inside the cell. So gut microbes synthesize ammonia from urea, and the host cells take it up. It likely carries very little deuterium because it's a gas. That's my big assumption. This just shows you that the ammonia availability depends upon the pH of the environment. This is at a low pH of 5, and over here is a high pH of 8, which the matrix is almost at 8. Its pH is high. And here you get practically none of the ammonium coming out as ammonia gas. But as you go up in pH, this increases. So you have almost 6.6% of the ammonium dissolved in the blood, in the matrix, is available as ammonia gas. And that can become important as a source of low deuterium protons to push into the intramembrane space. Mitochondria exploit small gases at pH 7.9, which is where the matrix sits. Nearly 6% of the ammonia in the mitochondrial matrix is a gas. Aquaporin 8 is available at that inner membrane to actively pull the ammonia across that membrane into the intramembrane space. And then enzymes in the matrix combine the remaining NH4 plus with alpha ketoglutarate to synthesize glutamate, and then add another NH4 plus molecule to make glutamine. And so that's a way to capture the remaining NH4 plus to keep the levels low so that it won't be toxic. Aquaporin 8 transports water, hydrogen peroxide, and ammonia across that inner membrane. So it can pull in all of these things that are going to be low in deuterium. So specialized transporters distribute ammonia, but not ammonium. And in fact, the RH factor proteins in the red blood cells, I did not know this until recently, they actually can import ammonia, but not ammonium, into the red blood cell and also out of the red blood cell. So it functions as a high efficiency ammonia transporter with the red blood cells carrying the ammonia, which can then be delivered to the liver. And those RH proteins have a channel that's highly selective for NH3, but not NH4+, which implies that ammonia is going to be low in deuterium. The red blood cells deliver it to the liver for conversion to urea. And then epithelial cells lining the gut express other RH proteins, but different ones basilaterally and apically that actually pull the ammonia out of the gut lumen that the microbes are producing from the urea. And they pull it out as NH3, leaving behind NH4+, therefore getting a source of low deuterium protons. Ammonia in the liver, it readily crosses the hepatocyte membranes, and especially in the alkaline environment, that's where you can get a lot of gas where it's quickly trapped. And then periportal hepatocytes possess high-capacity enzymes that convert it to urea, which is then transported to the kidneys and to the gut microbes. Paravenous hepatocytes act as a secondary high-affinity scavenging system. So they take whatever's left over and they convert it into glutamine. So as a way to keep the ammonia levels low in the blood, which will be toxic. So here's this image, a figure from this paper of the enterohepatic urea cycle, where the ammonia returns to the liver via the portal circulation, and then NH4 plus is passed out through the species. So you can see here's the colon, here's the ammonia, urea that's getting converted to ammonia. And of course, that becomes NH4 plus, but then the ammonia component of it, the gas component, goes back to the portal circulation. So the body is capturing the ammonia and letting the ammonium go out to the feces, which makes a lot of sense. So ammonia processing in the kidneys, ammonia is synthesized from glutamine in the proximal tubule over here. These guys synthesize ammonia, they release it into the lumen. This channel here is going to eventually become the urine. And over here, you can see all these guys are NH4 plus, not NH3. That's what's coming out in the urine. And the ammonia is being, a lot of ammonium is dumped here through this process of converting it from a glutamate and glutamine. And then it's carried in here, and then it's picked up and brought back in along this part of the chain by these cells that are in this ascending limb. The ascending limb, thick ascending limb cells push ammonia out into the interstitial space, interstitium into the collecting duct, out of the interstitium. Sorry, these guys put it into the interstitium, and then later on, the NH4 plus is preferentially taken out, goes back into here, and that's why this ends up as NH4 plus. Meanwhile, all this NH3 that's hanging out in here gets picked up by this. This is very highly fused, has a lot of blood vessels inside this area, where the red blood cells can soak up the NH3, and then leaving behind the NH4 plus that goes back into here and goes out. Very complicated process that goes on in the kidneys, which looks like it's trying to keep the NH3 into the circulation and dump the NH4 plus into the urine. Hydrogen sulfide and dimethyl sulfide, my last topic. The big picture, hydrogen sulfide easily crosses the plasma membrane and the mitochondrial membrane. Cystathionine gamma lyase and cystathionine beta synthase produce hydrogen sulfide from homocysteine and cysteine, primarily in the cytoplasm. And it's consumed in the mitochondria, yielding sulfate and thiosulfate. It's basically metabolized. And so probably depleted protons are being dropped off in the mitochondrial water through this process from the H2S. Methane thiol is derived from methionine, and it's metabolized by cell NBP1 to produce many useful likely depleted products. Exogenous administration of hydrogen sulfide reduced inflammation, promoted mucus production, and restored the microbiome in a mouse model of inflammatory bowel disease. Dimethyl sulfide is produced by microbes in shallow seas, and it seeds clouds and doesn't carry two depleted methyl groups, 6 depleted protons. Dimethyl sulfide oxidizes to methylsulfonomethane and dimethyl sulfoxide, both of which are commonly used therapeutically by naturopaths. And so here's a paper: hydrogen sulfide protects from colitis and restores intestinal microbiome and mucus production. Too much hydrogen sulfide, of course, is toxic, but there's a sweet spot where it's actually useful, and people are using it in low doses as a therapeutic option in the colon to reduce inflammation, restore the microbiome, and increase the production of mucus granules. So NBP1 and methane thiol, very interesting story here. Methane thiol is actually a volatile breakdown product of methionine, and the gut microbes produce lots of it, amounts of methane thiol. And all the products of cell NBP1 metabolism of methane thiol are likely to be excellent sources of depleted protons. And insufficient activity of this protein leads to bad breath due to dimethyl sulfide in the breath. Glyphosate is a copper chelator that could compromise the supply of copper to cell NBP1. And that would be problematic because then you would not be able to produce these very useful metabolites by breaking down the methane thiol, which goes to hydrogen sulfide plus hydrogen peroxide plus formaldehyde. All three of these are likely to be deuterium depleted. And so they will be useful molecules for the host. So this protein is called selenium binding protein 1. It's a methane thiol oxidase, and it catalyzes the conversion of methane thiol to these important metabolites. And it requires both copper and selenium as cofactors. Deficiencies result in bad breath and due to dimethyl sulfide. And then this is really fascinating. Dimethyl sulfide, the climate gas you've never heard of. This article is I have a lot of references here. Dimethyl sulfide is a small sulfur-containing gas that plays a significant role in climate change. So it decreases the amount of solar radiation that reaches the Earth's surface by inducing cloud formation. It's synthesized from this other molecule, dimethyl sulfonyl-propionate, DMSP, by marine phytoplankton in the shallow ocean water. So you think of the Mississippi River, and you think of all the glyphosates that's accumulating in the Mississippi River from all the farming that goes on upstream, and it pours the glyphosate out into the shallow seas at the mouth of the Mississippi, and that's going to affect this microbe, Emiliania huxley, which is an important marine phytoplankton species that synthesizes DMSP. And the reason why I say this is because I found a paper about glyphosate and its effect on this important microbe in the shallow seas. When they were exposed to glyphosate at two different levels, 36 micromolar and 360 micromolar concentrations, they got clobbered. And so this is showing here the regular control microbes, and then the low levels of glyphosate and the high levels of glyphosate here. And you can see they get the high levels of glyphosate just completely wipe them out. And they were also reduced by the lower level of glyphosate. So suppression of the release of DMS from phytoplankton in the shallow seas may have a significant impact on climate change through insufficient cloud formation over the oceans. So in summary, I hypothesize that human metabolic policy makes heavy use of small molecular hydrogen-containing gases to fractionate out deuterium to support mitochondrial health. The hydrogen-producing gut microbes lead to the production of essential depleted micronutrients, mainly the short-chain fatty acids and the methyl groups in methylation pathways. Inflammation is associated with many chronic diseases and conditions, but is this a way to deliver low deuterium hydrogen peroxide to the mitochondria? The complex treatment of ammonia at the renal tubules supports retention of deuterium-depleted ammonia in the circulation and passing out NH4 plus through the urine. Sulfur-containing gases, hydrogen sulfide and dimethyl sulfide, play interesting but poorly understood roles in biology. And thank you so much for your time.